Dear students, myself Dr. Zubari Arham Javed, lecturer in Botany Department of Microbiology, BOTN 4128. Today we will discuss use of physical methods in microbial growth control in our lecture number 25. As you all know, we have freeze drying, the section from the previous lecture, and we also discussed some other physical methods. Dear class, this portion is taken from your course outline, Control of Microorganisms. First of all, we will discuss about contents. The low temperatures methodology of heat control means control of microorganisms by heat methods, and which we discuss the low temperatures methodology. Then the filtration, which includes in the mechanical control or mechanical methods in microbial growth control. And in filtration, we discuss filtration of liquids and the filtration of air. In filtration of liquid, we discuss about types of filters like depth filters and the membrane filters. While in filtration of air, we discuss about surgical masks, different cotton plugs and the HEPA filters. Here you go about low temperatures, in which we have three main methods like freezing, like refrigeration and the freeze drying, which also called the lyophilization, and okay, class. Although our emphasis is on the destruction of microorganisms, often the most convenient control technique is to inhibit their growth and reproduction by the use of either freezing or refrigeration or lyophilization or freeze drying. First of all, we discuss the freezing. This approach is particularly important in food microbiology because freezing items at -20 degrees centigrade or lower stops microbial growth because of the low temperature and the absence of liquid water. Some microorganisms will be killed by ice crystal disruption of cell membranes, but freezing does not destroy all contaminating microbes. In fact, freezing is a very good method for long-term storage of microbial samples when carried out properly, and many laboratories have a low temperature freezer for culture storage at -30 or -70 degrees centigrade. As you see here, here are some ultra low temperature freezers which are almost available in all our labs, in our hospital laboratories, in our university campuses. These are widely used for storing microorganisms, microbial samples for longer time. Second one is refrigeration or refrigerators. Pure cultures can be successfully used to control the microbial growth at 0 to 4 degrees centigrade, either in the refrigerators or in cold rooms. Dear class, you can easily watch a number of cold rooms or refrigerator rooms in your nearby hospitals, in your nearby laboratories or industries. Because frozen food can contain many microorganisms, it should be thawed in a refrigerator and consumed promptly in order to avoid spoilage and the pathogen growth. Refrigeration greatly slows microbial growth and the reproduction, but does not halt it completely. Fortunately, most pathogens are mesophilic and do not grow well at temperatures around 4 degrees centigrade. Refrigerated items may be ruined by the growth of psychrophilic and the psychrotropic microorganisms, particularly if water is present. Thus, refrigeration is a good technique somehow only for shorter term storage of food and other items. Here you have the graphical presentation of ultra low temperature freezers. As I already told, that these are easily available in the laboratories of our campuses and also available in the small test laboratories. Next one is freeze drying and lyophilization. Freeze drying or lyophilization is the drying of a material from the frozen state. This process is used in the manufacture of some brands of instant coffee. Freeze-dried instant coffee has a more natural flavor than other kinds. Dear class, freeze drying is also known as lyophilization, and microbiologists use lyophilization for longer term preservation rather than for destruction of cultures of microorganisms. If we discuss about the procedure, then what will we do? That organisms and vials, first of all, the organisms and vials are rapidly frozen in the presence of alcohol and the dry ice or in liquid nitrogen. Then it's subjected to a high vacuum to remove all the water while our specimen is in the frozen state, and then we sealed under the vacuum our specimen finally. Rapid freezing allows only very tiny ice crystals to form in cells so the organisms survive, and this process organisms so treated can be kept alive for years, stored under vacuum in the freeze-dried state. As you also see the freeze-drying process from this graphical presentation, that first we freeze rapidly, then evaporation of ice and we remove all the liquid, then pure dry bacterium. We achieve the pure dry bacterium, which we call that our specimen or the sample is fully lyophilized. And then when we add water to a dry product, it is restored to its original condition. Class, here you go. Here you have a stopping tray dryer, in which the trays supply the heat needed to remove moisture, and after completion of lyophilization, or means about 24 hours, the device automatically stoppers all the vials that we placed in it, and it is a manifold dryer in which pre-frozen samples in vials of different sizes can be attached through ports into the dryer, which supplies, as you all saw these ports, which supplies a vacuum to remove water. The sample will dry in 4 to 20 hours depending on its initial thickness. Unless the tray dryer, this device allows samples to be added and removed. Unlike the tray dryer, this device allows samples to be added and removed. Now class, here is our third portion, that is the mechanical removal methods. Here we skip our second portion, which is the chemical removal methods. We will cover all the chemical methods or the removal methods in our next lectures. Today we will discuss the mechanical removal methods, in which we have filtration method. If we see the filtration, we will discuss filtration by liquid and the filtration of the air. Filtration. Filtration is an excellent way to reduce the microbial population in solutions of heat sensitive material, and sometimes it can be used to sterilize solutions. If we see the definition, then filtration is the passage of a material through a filter or screening device. Filtration has been used since Pasteur's time to separate bacteria from media and to sterilize materials that would be destroyed by heat. Dear students, rather than directly destroying contaminating microorganisms, the filter simply removes them. It is the best method to remove the microorganisms, either to try to destroy all the contaminating microorganisms. There are two ways of filtration, like filtration of air and the filtration of liquid. If we discuss about the filtration of liquid, then we have two types of filters, the depth filters and the membrane filters. Control the growth by filtration requires filters with exceedingly small pores, and both type of these filters have exceedingly small pores. If we discuss about depth filters, then depth filters consists of fibrous or granular material that have been bonded into a thick layer filled with twisting channels of small diameter. The solution containing microorganisms is sucked through this layer under vacuum, and microbial solutions or microbial cells are removed by physical screening or entrapment, and also by adsorption to the surface of filter material. If we discuss about the composition of the depth filters, then we see that these type of filters are made of diatomaceous earth, which we also call Berkfield filters, and also these are made up of unglazed porcelain, asbestos or some other similar materials. Class, unglazed porcelain is also known as Chamberlain filters. And if we discuss about membrane filters, the membrane filters have replaced depth filters by many purposes, because these circular filters are porous membranes, a little over 0.1 millimeter thick. If we discuss about the composition, these are made of cellulose acetate, cellulose nitrate, polycarbonate or other synthetic materials. Pore size, although a wide variety of pore sizes are available, membranes with pores about 0.2 micrometer in diameter are used to remove most vegetative cells but not viruses from solutions ranging in volume from 1 ml to many liters. Membrane filters with depth filters. The membranes are held in special holders, as you see in the figure, and are often preceded by depth filters made of glass fibers to remove larger particles that might clog the membrane filter. About the working, the solution is pulled or forced through the filter with a vacuum. Here is your vacuum. Or with pressure from a syringe, peristaltic pump, or the nitrogen gas bottle, and collected in previously sterilized container. Here you see that the filtrate solution is collected in this beaker. This is a complete filtering setup. The non-sterile solution is in the Erlenmeyer flask. Here is Erlenmeyer flask. And then a peristaltic pump or the vacuum pump is here. And this is the forced solution through the membrane filter unit. And all the solution after filtration is collected in this beaker. Here is a schematic representation of the membrane filtration setup that uses a vacuum pump to force liquid through the filter. The inset shows a cross section of the filter and its pores. Like this is the filter and how these pores channels and how the liquid is cross the membrane through these pores channels, which are too small for microbes to pass through. This is the cross section of a membrane filtration unit. These are the torapore filters and these are the filter spores. This is the filling bell and this is the vent. Several membranes are used to increase its capacity, and if we see the uses, it is widely used to sterilize pharmaceuticals, ophthalmic solutions, culture medias, oils, antibiotics, and other heat sensitive solutions. Second one is filtration of air or air filtration. Air also can be sterilized by filtration, and the two common examples are surgical masks and cotton plugs on culture vessels that let air in but keep microorganisms out. Other important examples are laminar flow biological safety cabinets, which employ high efficiency particulate air filters. A type of depth filter, which is commonly known as HEPA filters. These are able to remove 99.97% of 0.3 meter particles. Here is laminar flow biological safety cabinets, which is also known as HEPA filters. Laminar flow biological safety cabinets or hoods force air through the HEPA filters, then project a vertical curtain of sterile air across the cabinet opening. Here is the cabinet opening. This protects a worker from the microorganisms being handled within the cabinet and prevents the contamination of the room. Here you see a technician pipetting potentially hazardous material in a safety cabinet. And here this is a schematic diagram showing the airflow pattern, that how the air is moved within the high efficiency potential safety cabinets. A person uses these cabinets when working with dangerous agents such as Mycobacterium tuberculosis and the tumor viruses. They are also employed in research labs and industries such as the pharmaceutical industry when a sterile working surface is needed for conducting assays, preparing medias, and examining tissue cultures and the likewise. These HEPA filters are very helpful while doing all these works. So class, this is all about your today's lecture, in which we are studying about the freeze drying or lyophilization, and then we discussed about the mechanical removal methods like filtration of the air or filtration of the liquids. Class, these all data are collected from your reference books, and these are mentioned in this slide from the Surinder Kumar textbook of microbiology, from the microbiology principles and exploration, and from the essential microbiology. Class, if you have any question, then you are freely ask me in your Google Classroom. Thank you so much.